Bismillahirrahmanirrahim. Assalamu alaikum. How are you, students? I hope you all are fine. God bless you. Today we will learn about drawing for class 6 scientific diagram. Okay, students, you listen to me carefully. Keep practice copy and stationery along with you. Take a screenshot of every slide which will be shared over here. And one more thing, as you know, you do not have drawing copy in your copy set. So please make a portion in size copy at the back side of pages 20 to 25 to draw the diagram. Okay, draw all diagram step by step to the instruction. Before starting, make a title page of scientific diagram. I hope you understand all instruction carefully. Okay, students, you should know the rules of scientific diagram. What are these rules? Okay, let me share these. These are show on the screen rules for scientific diagram. This is the first rule is draw neat and clean diagram in the center of the page. It means you draw neat and clean diagram because neatness and cleanness is the very important for making drawing. So in the end you draw in the center of the page and leave equal distance around the page. I think you understand first point. Now move second point, use sharp and pointed pencil. Sharpness and sharp pencil and pointed pencil is very important for making drawing of scientific diagram. Okay, now point number next point is label the diagram at right side of page with sentence style. Means you draw label all the time right side of the page with sentence style because this is very important. The first letter of the labeling is capital and other are small. Always draw a box around the picture. Okay, when you will complete your diagram, you will draw a box around the picture. Okay, next point, do not use any pen, marker or color on the drawing page. Only use pencil. It is very necessary you will use only pencil. There is no need of pen, marker or color for scientific drawing. Scientific instrument drawing do not need any labeling. Just you will write the name bottom side of the diagram. Next, write the name of diagram at the bottom of drawing. When you when you will complete your diagram, you draw the write the name bottom side of the diagram. Okay, I hope you understand all the point of how we draw a scientific diagram. If and you can easily draw in your practice copy. Can I repeat again these points? Okay, now we will move further. Today we will draw three scientific diagrams: measuring cylinder, burette, and plant cell. We will draw these diagrams one by one. Measuring cylinder. It is used in the lab for the measurement of liquid. We will draw this diagram step by step. Okay. You will practice this diagram by step. Okay. This is the steps of how we draw the diagram. Draw two parallel lines of 8 cm with the distance of 2 cm. Observe carefully. You will draw the one line 8, by the help of scale 8 cm. Another line in front of this line. Both lines are 8 centimeter. These two lines are called parallel line, and the this measurement of these lines are 8 centimeter. Okay, and the distance between these two lines are 2 centimeter. Okay, okay. You you draw in first these two lines. Now next step, at the top make the head of measuring cylinder with its beak. Observe carefully. At the bottom, close the cylinder and draw base of 3 centimeter with the distance of 1 centimeter. Okay, this is the head and bottom close by the line, and then you draw a beak like this. Okay, again I will show how you will draw this diagram. Close upper and lower side of the 8 centimeter line, and then draw a base, the base 3 centimeter, and join the line by the cylinder and make a beak. Okay, now next step, draw a small line on the cylinder for measuring the scale. Observe it. Draw a box around it. In first we make a measuring scale on the measuring cylinder. This is very important because we measure the different types of liquid by the cylinder, and make and draw a box around it. Okay, I hope you understand all steps and you will draw easily in your copy. This is the actual diagram which is shown in front of you. Okay, now we will move another diagram, burette. Burette is also used for measurement of liquid in the lab. How we draw? We draw step by step. Step number 1, draw two parallel lines of 7 centimeter with the distance of 1.5 centimeter. Like measuring cylinder, we will draw two parallel lines like this. And after 7 centimeter, make it more narrow with the distance of 0.5 centimeter. This is narrow tube is of 3 centimeter long. This is the one parallel line 7 centimeter, and another line it is parallel to the distance of The two line is 1.5 centimeter, 7 centimeter, and the distance is 1.5 centimeter. Make another two lines 3 centimeter distance. Observe a 3 centimeter line and then move further. Draw a nozzle at the end of the narrow tube by small oval shape like this. Can you observe it? Okay. Now draw a measurement of burette as shown. Like measuring cylinder, we will draw a measuring scale on the upper side of the tube. Okay. This is the actual diagram. Observe it and draw in your copies. I hope you understand all the steps of burette. Now we will move towards the third diagram. This is plant cell. Plant cell is a very important cell in your syllabus. We will draw this diagram by using microscope. This is we will not see by the help of naked eye. This is microscopic structure. How we draw it? In first you observe the diagram, and then we will draw step by step. Draw a rectangle shape for the cell wall of plant cell. Observe the diagram. In first we draw the first layer of the plant cell. This is thick wall. This is called cell wall. Okay. In first we draw a rectangle shape for the cell wall. Okay. Like this we will draw inner line to make the cell membrane. Okay. This is the inner line. This is very thin, little bit thin than cell wall. This is called cell membrane. Can you observe it? Okay. Now we will move another step. In the center, this is the center part of the cell. Draw a large size irregular shape for plant vacuole. This is a plant vacuole. It is large in size in a plant and it is in the center. Again, in the center of it, draw a small circle. You draw a small circle, and this is a nucleus. Okay, this is the small circle, and it is near than the cell wall or cell membrane. And observe different oval shaped structure. You will see the green color oval shaped structure. These are called chloroplast. These chloroplast is very important for the plant cell because they are prepare their food by the help of photosynthesis. Okay. Now another oval structure. Observe it. This is called mitochondria. These are the small vacuole, chloroplast. These are the small oval structure, chloroplast and mitochondria. mitochondria for cell organelles and the label them accordingly. Okay, and first we draw all the structure like the picture, and then we label all the parts of the diagram. If you will take the screenshot of the diagram, you will easily draw it. Okay, now I will repeat again how we draw the cell wall. In first we make a rectangle shape like this, and the outer layer is called cell wall. Then we draw a thin layer. This is called cell membrane. After that we draw a inner line in the center. Draw a large size vacuole like this. This is a large size vacuole, and again in the center of draw a small circle. We draw a small circle. This is called nucleus. And then you draw a small oval structure. These are called chloroplast. These all are organelles of the cell. Now, another oval structure is called mitochondria. After that, you will label the diagram and make a box around the picture. Okay, and then you draw the box around the picture. Okay, now again I will explain how we draw the different steps of measuring cylinder. Okay, I will explain one by one. Draw a two parallel line of 8 centimeter with a distance of 2 centimeter. This is one line. We will take a scale and measure the 8 centimeter line. In front of this line, or we can say that the parallel line, again we draw another line. This is called these two parallel lines. Okay, the distance between the two lines is 2 centimeter. This line is 8 centimeter, and the distance between the two lines is 2 centimeter. Okay, I think you follow this step. Now we will move another step. At the top, make the head of measuring cylinder with its beak, and at the bottom close the cylinder and draw base 3 centimeter with the distance of 1 centimeter. Like this, we close the two lines, upper side and lower side, and make a base 3 centimeter and join these lines by the end point of the cylinder. Okay, now this is the beak of the cylinder. We will easily draw step by step. Now move further. Draw small lines on the cylinder for the measuring the scale, and draw a box. Okay, these are the small lines on the cylinder for the measurement, because the measurement is very important, because we measure the liquids in use into the lab. Then 20, these are the scales. Okay, to use the scale by the help of meter. Draw draw a box. Okay, draw a box outside of the cylinder. Now move to the, this is the actual picture of the measuring cylinder. Okay, understand? Now burette. How we draw the burette? Step number one is same like a measuring cylinder. Draw two parallel lines of 7 centimeter with a distance of 1.5 like this. After 7 centimeter, make it more narrow with the distance of 0.5 centimeter. This narrow tube is of 3 centimeter long. Observe these two lines. These are the 7 centimeter lines, and distance between the two lines is 1.5 centimeter. And then you will draw a narrow line. Narrow two lines. These lines are 3 centimeter, and the distance between two lines is 0.5 centimeter. Okay. Draw a nozzle. This is a nozzle. We will drop the liquid from the and to control the liquid. We will make a stopper. Observe the diagram. This is stopper. Draw a measurement on burette as shown. Draw a box. These are the measurement which we measure the different liquid by using burette. Okay. This is the actual picture of the burette. Again, plant cell. And first you observe the diagram and then you draw step by step. Draw a rectangle shape for the cell wall of plant cell. Rectangle shape you draw. This is the first layer you draw in first by find, find out the shape or structure of the cell. Okay. Then draw inner line to make the cell membrane. This is inner line. Observe carefully. We draw another line. This is little bit thin for the cell wall. This is cell wall, and this is inner layer is called cell membrane. And then in the center, draw a large size vacuole. This is the center of the cell, and we draw a large size vacuole. Okay, and then draw a center of it. Draw a small circle. This is the small circle, and the small circle is called nucleus. It is near them to the cell membrane or cell wall. Okay, and then you draw a small ovule like this. These are the green oval structure. These are called chloroplast, and these are very important in the plant cell. Another oval structures are these. These structures are called mitochondria. Okay, and then you make a box. Label the diagram, and then you make a box around the diagram and write the name at the bottom side. Okay, now now home task. This is your home task. I think you observe and draw easily after understanding these steps. So you, I mean, draw a neat and labeled diagram which we learned here. Draw neat and labeled following scientific diagram at the back of your science copy. Measuring cylinder, burette, plant cell. Make a portion of scientific diagram at the back of your science copy. 20 to 25 pages. I think all of you understand all the home task. Okay, now again I will repeat your home task. Draw neat and labeled diagram at the back of your science copy, measuring cylinder, burette, and plant cell. Make a portion of scientific diagram at the back of your science copy, 20 to 25 pages. Okay, I hope you understand all homework. It's all for today. Thank you.